Howdy, Aztecs. Welcome to Module 2, and this is Module 2A. It's continuing Chapter 2, so if you look at the Module 1 stuff, there was some Chapter 2 information in it. But now we're in Module 2A, Chapter 2, but now we're going to talk about biochemistry. And biochemistry has 3 parts, so biochemistry 1, 2, and 3. Anyway, here's our periodic table of elements. Do you remember from the previous videos? God, remember the elements that we said make up God? If you said chone, you were right. Carbon, hydrogen, oxygen, and nitrogen. Chone. And then sometimes, it depends on the book, they'll put in a P, phosphorus, and a sulfur. So chones P or chone PS, however you like to do it. But anyway, those are the really important elements in life. So this cartoon shows organic versus inorganic molecule structures. So you've probably heard the term organic bandied about in the culture. Oh, I only eat organic, or is that organic beef? Because I only eat organic beef. Well, it takes a different understanding of organicity in biochemistry. So if you are an organic molecule, this is tricky. You have carbon in you. You've probably heard that. If you're organic, you have carbon in you. But you also have hydrogen in you. Because check it out. These guys are all organic. So ethane, propane, hexane. These H's are in the wrong spot. So these black circles, those are all carbons. And then the smaller circles are all hydrogens. So if you are an organic molecule, you have carbon in you and hydrogen. If you are an inorganic molecule, you have other elements in you. Check it out, though. This is where it gets tricky. This is the carbonate ion, so it does have carbon in it, but it doesn't have hydrogen as well. So that means it's an inorganic molecule, not an organic molecule. And then here's some more inorganic molecules. Everybody's probably heard of ammonia, and I know everyone's heard of water. Those are inorganic molecules or compounds. Inorganic compounds have no carbon. Well, there's the exception to the rule right in front of us with the carbonate ion. And of course, there's always an exception to the rule. So in general, inorganic compounds tend to not have carbon. They tend to be small and simple if you're an inorganic compound, like water's pretty simple, H2O. Salt is pretty simple, sodium chloride. And then some acids and bases like HCl, hydrochloric acid. Organic compounds, like I said, they contain carbon, and most of the time, remember there's an exception to every rule, but most of the time organic compounds contain hydrogen as well. Most organic compounds are large and they're covalently bonded. So do you remember what a covalent bond is from the previous videos? Well, you can pause this and think about what a covalently bonded molecule is. If you said a covalently bonded molecule is when two elements or two atoms share electrons, you would be right. Those include, organic compounds include carbohydrates, lipids, proteins. So all the things we like to eat are organic compounds. And nucleic acids, that's DNA and RNA. Organic compounds often contain these things called functional groups. And if you've had chemistry, then you know functional groups. But if you haven't had chemistry, these are functional groups. This is a cartoon of all the functional groups that we're interested in anatomy and physiology. So a functional group is a portion of a molecule that is classified as a group of bound atoms. So for example, in highlighted in gray, CH3. So oftentimes in the chemical world, you will find this CH3. That is a functional group. And then this R, that's just that's the bigger molecule that the functional group is attached to. So every one of these in the cartoons have this R group, and it's just it could be any molecule, because the functional groups can attach to a lot of different molecules. In organic chemistry, which is mostly what we're interested in anatomy and physiology, it's really common to see molecules that have a carbon backbone, so carbon, 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 with functional groups attached to that carbon backbone chain. So let's look at some more functional groups that are interesting for us. Definitely a methyl group is interesting for anatomy and physiology. Carboxylate is really interesting because that is attached to proteins. Hydroxyl, those are everywhere. Acetyl groups, those are important in A and P. Amino groups, that's attached to the other end of a protein, or if you have a single amino acid that makes up, so amino acids make up proteins, but at the end of a single amino acid or a big protein chain, you'll have an amino group and you have a carboxylate group. Sulfhydryls are interested in anatomy and physiology. Phosphoryl groups, so you can see the phosphate here. But now I ask a question, which functional groups can hydrogen bond? So do you remember what a hydrogen bond is from the last videos? Well, it's a bond that is formed between two molecules, and one of the molecules has a polarized hydrogen that's partially positive, and the other molecule has a electronegative atom like oxygen or nitrogen, for example. So which of these functional groups can form hydrogen bonds? Well, this one could, carboxylate. The hydroxyl group can, because look, this would be a covalent bond here between the hydrogen and the oxygen, but it would be a polar covalent bond. That means this hydrogen would have a partial positive charge, and if it came in the vicinity of this partial negative charge on this oxygen, then you could form a hydrogen bond. And remember, the hydrogen bonds are formed, the way they draw them is they usually draw these little dashed lines to show that there's a hydrogen bond. And you can pick out the rest of the functional groups that you think could form hydrogen bonds. Okay, and then I put the blue boxes around the functional groups that are important for us. Okay, so here's a definition of the hydrogen bond. It's a free hydrogen. Hydrogen bond can be formed between a free hydrogen or a hydrogen in a polar molecule. So for example, this hydrogen is in a polar molecule because one pole, the oxygen, where the oxygen is, that's the negative pole, and where the hydrogen is, that's the positive pole. So this would be a polar molecule, or it would be a functional group part of a polar molecule. So where the hydrogen, partially positive hydrogen in a polar molecule, is attracted to the negative portion of another polar molecule. So this would be another polar molecule, because remember, if you have an oxygen or a nitrogen, those are really strongly electronegative, and they'll form covalent bonds, but they'll kind of hog the electrons, so that makes that a polar molecule. Hydrogen bonds are really important for your future in taking classes anyway. So here's an example of a question. Hydrogen bonding is present in which of the following functional groups? The hydroxyl group, carboxylate. Remember, there's a nitrogen. That means it's forming a bond with two hydrogens here, but it's electronegative, so this will be a partially negative nitrogen. The phosphate or phosphoryl group. Here's some oxygens that are negative. So the answer would be all of the above. All of those can form hydrogen bonds. Water. Oh, wow. There's whole classes just on water and molecule water. But for us, water and living organisms, that's what we're going to look at. The most abundant inorganic compound in a living organism is water. You probably have heard this. 60 to 80% of the volume of your cells is water. It's not only the most abundant inorganic compound, it's the most important inorganic compound to us. As you know, we all need water. And water is important due to water's properties. So properties of water. It has high heat capacity. That means it can absorb a lot of heat but not change its temperature very much. It can release a lot of heat, but its temperature won't fluctuate very much. So that prevents sudden changes in temperature near oceans, for example. So climates are always really nice near large bodies of water. And we use water as evaporative cooling. So we sweat, the water evaporates, pulls the heat from our body. Water has polar solvent properties. That means water can dissolve and dissociate ionic substances. So what's our famous ionic substance that we talk about all the time that has an ionic bond? Positive sodium, negative chloride. They're attracted to each other, forms sodium chloride. That's salt. Reactive properties of water. So water takes part in chemical reactions. It's a necessary part of hydrolysis reaction. That's when you break the bonds in molecules. And then dehydration synthesis reactions. That's where you pull water out of the molecule. You put water into a molecule by pulling. So hydrolysis breaks bonds. Dehydration synthesis makes bonds. Okay, we've looked at this cartoon before. Remember, there's intermolecular bonds. So these are polar covalent bonds between oxygen and hydrogen. And then intra, or between molecules, you have the hydrogen bonds. Reactions that require water to break bonds are hydrolysis reactions, and we have a cartoon for that in the next slide. So up to, I gave you some stats on the previous slide. I think it said 60 to 80% of the volume of a cell is water, but it can get up to. So if you have a cell that's super hydrated, 80% of the volume inside that cell could be water. Water helps digest your food. It breaks down biological molecules. And then we talked about these on the previous slide, the vital properties. All right, these reactions are really important going forward in taking your classes. Hydrolysis reaction. So so just a couple of definitions. This could be this cartoon. This could be a cartoon of a protein or a carbohydrate. So if it were a cartoon of a protein, these monomers or single things would be amino acids. If it were a carbohydrate, these monomers could be like glucose. So if monomers are linked, and it just shows two monomers in this chain, but you can have a really long chain of monomers, and then that long chain of monomers would be called a polymer. So in this cartoon, these monomers are linked by a covalent bond here. If you want to break that bond, you can do this thing called a hydrolysis reaction, where you put a molecule of water in here into the bond and it breaks it. So remember, you've got two H's and one oxygen in water. So here's one of the H's. Here's the other H that you used from the water, and here's the oxygen that you used from the water. And this oxygen here was not originally part of the water molecule. This oxygen was from here in the monomeric. So on the reactant side, or this is the reactant side, and then this side is the product side. Hydrolysis reactions. Polymers are broken down into monomers through the addition of water molecules across the bond. Breaks the bond. The opposite of a hydrolysis reaction is a dehydration synthesis. So check it out. Same thing. This could be a protein or a carbohydrate. You've got a long string of monomers. It's called a polymer. But between now this is not a polymer though. This is a single monomer on this side. Here's a single monomer on the reactant side. But look, on the tail end of this monomer 1 is an OH. Monomer 2 also has an OH, but they just highlighted the two H's and an O, because what you're going to do is you're going to take water out between these two monomers, and here it is in the reaction on the arrow. Water is leaving, and that just leaves this oxygen here, and then that oxygen is left to form this covalent bond. So a dehydration synthesis, this kind of makes sense. You're dehydrating, so the water is leaving, and synthesis means you're creating something new, so you're putting monomer 1 and monomer 2 together. Patterns of chemical reactions. So there's lots of different chemical reactions, and this is not a chemistry class, but we kind of have to go over them in general. There's synthesis reactions in chemistry, or combination reactions. So synthesis just means to synthesize or make something. But in chemistry, in the synthesis reactions, you could be a decomposition or an exchange reaction. So synthesis reactions take this basic form. You have A, so on the reactant side you have reactant A plus reactant B go together to make product AB. So in a synthesis reaction, in general, atoms or molecules combine to form a larger, more complex molecule. That's AB. Synthesis reactions always involve bond formation. So in this cartoon, you're making a bond. So for example, a synthesis reaction could be an anabolic reaction when you're joining two amino acids together. So like we showed in the previous slide. You could be a decomposition reaction where you break apart this is the reactant AB, and you break reactant down into two products, A and B. And then in exchange reaction, you have AB plus C on the left side of the arrow and AC plus B on the right side of the arrow, and then you just switch them around. So let's look at the cartoon over here. So in this exchange reaction, this is specifically a replacement or double exchange reaction, and you can see you begin with AB plus CD, so those two A and B, C and D are together, and you just swap out, and A goes with D to make AD, and C and B go together to make CB. Okay, and then here's some more cartoons. This just shows an exchange reaction with an example in it. So here's glucose. Here's a glucose molecule, C6H12O6, plus ATP, or adenosine triphosphate. And you just take one of these phosphates from ATP, and you put it on the glucose molecule. Now in the chemical reaction, it shows the arrow going down. Usually we've been seeing in chemical reactions, you have the reactants on the left side of the arrow and the products on the right side of the arrow. In this, the way they drew this, they have the reactants on the top of the arrow, and the products are down here. So exchange reactions can involve both synthesis and decomposition reactions. Bonds are made and broken. So it gets pretty complicated, and if you take a chemistry class, you can learn more about that. Oxidation reactions. These are really important. I even teach oxidation reduction reactions again in 202 and again in 205. And they're also taught again in 218. So you'll see these, might as well make them your best friend right now. So here's a molecule. Here's the purple molecule. And you can see it has an electron here. And then, and this guy is going to be the electron donor. Now, the terminology gets really tricky. This guy, purple guy, is going to be the electron donor. The green guy, this green box here, this guy's going to be the electron acceptor. So when the purple guy donates his electrons, you don't want free electrons floating around in your cells. You want another molecule to be there to accept it. So electron donor gives his electron to the electron acceptor. Now, this guy, the purple molecule, lost one negative charge, so it's going to become more positive overall. Its charge overall, if it lost one negative electron, that's one negative charge, it's going to become more positive by one positive charge. And that process of this molecule donating its electron is called oxidation. And then this green guy, he's going to get one more negative charge than he already had. So he's going to become more negative. His overall charge is going to become more negative. And that's where it gets a little confusing. But this guy that accepted the electron undergoes a process called reduction, which sounds weird. You're getting something, so this green guy is getting an electron, but he gets reduced. It sounds counterintuitive, but that's the way the game is played. Oxidation reduction reactions are reactions where food fuels are broken down for energy, and every time you see energy, just be thinking of electrons and ATP. Electron donors lose their electrons. We went over that, and they become oxidized. The electron acceptors receive electrons, and they become reduced. So in this cartoon, it shows C6H12O6, that's glucose, plus oxygen gives CO2, water, and ATP. So this glucose molecule gets oxidized because you're tearing off its hydrogens and it's accompanying electrons. So glucose is getting oxidized because it's losing electrons. And oxygen is actually accepting those electrons, so it gets reduced. What about the rate of chemical reactions? What are they affected by? How fast and how efficient can a chemical reaction be? Well, the rate of chemical reactions are affected by temperature. So if you increase the temperature if you increase the temperature such that your reaction, your enzyme is working optimally. If you increase the temperature within a certain range, you'll increase the reaction rate. So your enzyme can work really fast within a certain increase in temperature. But if you increase the temperature too much, then your enzyme will break down. If you increase the concentration of the reactants, so remember the reactants are here, and then the products are on the right side of the arrow. If you increase the amount of reactants here, oh sorry, let me put your enzyme here over the arrow. If you increase the concentration of the reactants to a certain degree, you'll also increase the rate of the chemical reaction. So that means you'll make more products quicker. If you have a smaller particle size, so if you've got smaller reactants, that can also increase the rate of the chemical reaction. And then this term is a catalyst. So a catalyst is something that changes one thing into another thing. So a catalyst will increase the rate of the reaction, but it won't change itself. And the best example in anatomy and physiology of a catalyst is an enzyme. So enzymes are biological catalysts. And then this is just a table in Marriott. It shows the factors that increase the rate of chemical reactions. I don't know if this is exactly true. But most chemical reactions are reversible. If you are a reversible reaction, you'll have a double arrow, like this guy right here. So this is saying water plus carbon dioxide gives carbonic acid. And then carbonic acid, when it breaks down, so it goes this way with the arrow, when carbonic acid breaks down, it makes CO2 and water. So it's a reversible reaction. Reversibility is indicated by the double arrow. We talked about that. When the arrows differ in length, the longer arrow indicates the more rapid reaction. So you could have an arrow that looked like that and then a little arrow that looked like that. It's reversible, but the reaction going to the right is way more rapid because the length of the arrow. If the reaction is neither going forward or reverse, then you have chemical equilibrium. Some biological reactions are essentially irreversible. Most chemical reactions are reversible, but some are irreversible. It depends on the energy requirements of the system and then how fast the products are being removed. Okay, endergonic and exergonic. So this is energy flow. All chemical reactions are either exergonic or endergonic. So exergonic reaction. So let's look at this. So you'd have energy on the y-axis here. And it says in an exergonic reaction, you have a net release of energy. So here's the reactants over here. Reactants, and then the products are down here. So the reactants have a lot of potential energy. So remember, potential energy is like when you're at the top of the hill, you're not moving, you have a lot of potential to release energy. So the reactants are up here, and you need to, in this cartoon, in exergonic reactions, you need to have some energy to get the reaction started. So that's this part right here, from here to here. You need to put in a little energy to get the reaction started, but once you get the reaction started, you go from this much energy to this much energy. So all this energy was the energy that was released in the reaction. And the products down here have less potential energy than the reactants. So remember, here's the potential energy of the reactants. Here's the potential energy of the products. And exergonic reactions include catabolic and oxidative reactions. So remember, we looked at those redox reactions where electrons are shuttled around, and then catabolic means breakdown, reaction that breaks things down. And then endergonic is a reaction, a major chemical reaction where you absorb energy. So again, Gibbs free energy is on the y-axis, and you need to have some you need to put in activation energy to get the reaction started. So you have to absorb, you have to absorb energy into your system. And here's the reactants over here. Here's the products here. And so if this is the product at this point, it's still got a lot of energy in it, right? Because you come over here and read energy off the y-axis. These products have a lot of energy, and it shows here, this is their potential energy. This is energy stored in the products. And anabolic reactions are an example of an endergonic reaction. Okay, chemical reactions and water. This is cool. So here's our beaker full of water. Here's our water molecule. And then here is a salt tablet, or a little salt rectangle. And that's sodium chloride, as you know. And then if you take this little piece here and you blow it up, here's the sodium ion, and then these big green balls are chloride ion. And then here's the water molecules over here in solution. And you can see that the partially positive hydrogen atoms in the water molecule are attracted to the negative chloride ion. So the water molecules surround this chloride ion. And then the partially negative oxygen molecules up here are attracted to this positive sodium ion. And that's how you dissolve salt, basically, is water circles each one of those ions. Water is the universal solvent because it can dissolve things. So if you are a solvent, water's not the only solvent, but if you're a solvent, you're a liquid or a gas, you can dissolve things, and those things you're dissolving are called solutes. So the salt, the sodium and the chloride and the salt would be the solutes. And you have a thing called a solution that forms when you have a solute of really tiny sizes are dissolved. So like salt dissolving in water, that would be a solution. You could also, if you have a solute that's a little bigger, so it's an intermediate size solute, and it dissolves, it's called a colloid, because the colloid then it's more translucent, so you can't exactly see through it. My favorite colloid, mayonnaise. All right, mixtures. Most matter, remember matter takes up space and has mass, they exist as mixtures. And a mixture is two or more components physically intermixed. Now, you probably learned all this in ninth grade in physical science. So 3 types of mixtures. There's solutions, we just talked about those. Colloids, we talked about those. And then suspensions, we didn't talk about those. So a solution in our table down here would be like salt water. So we looked at that. Sodium chloride and water. Particle type would be ions, for example, sodium and chloride ions. Particle size small, yeah, those are real small ions. Can the solution scatter light? Does it settle while standing, and does it separate by filtration? And all of these are no. If you are a colloid, so here's Jello, that's a good colloid too. The particle sizes are small clusters of ions, atoms, or molecules. The particle size is medium. And we don't really, this is kind of for a physics class, so I don't really care about that, but you can look at it if you want to. And then a suspension, an example would be muddy water. And the particle type in a suspension would be a large cluster. Particle size would be large. Another example of a suspension that's interesting for us in anatomy and physiology is blood. Blood is a suspension. Okay. So in this cartoon, it shows, here's the solute, so that could be sodium chloride like we've been talking about. Here's the solvent, that's water. You put the solute in the solvent, and you get a solution. So the solution is the mixture that forms when the thing dissolves in it. If you are soluble, that means you can dissolve in a liquid. If you are insoluble, you cannot dissolve in a liquid. Okay. True solutions. This is funny because are there false solutions? I'm not sure, but I guess. Concentration of true solutions can be expressed as a percent of the solute in the total solution. And the solution would assume to be water. So if you have parts of solute per 100 parts of solvent, then the units for that would be milligrams. So these would be the parts of solutes in milligrams, and 100 parts of solvents, that would be in deciliters. And then maybe if you've had chemistry, you've heard of molarity. So molarity is the same thing as moles per liter, and its unit is big M. So you have 1 mole of an element or a compound. So an element would be like a mole of sodium, or you could have a mole of water. That's a compound. Its atomic or is equal to its atomic or molecular weight in grams. That's what a mole is. Mole, and we're not really going to work with moles in anatomy and physiology. You probably saw them in chemistry if you have it. Types of solutions. So we've already talked about this. So here's a solution. Here's the properties of a solution. If you are a solute in a solution, then your size is less than 1 nanometer. So chloride and sodium would be, those ions would be less than 1 nanometer in circumference. The appearance of you, if you're a solution, you'd be clear. So when salt is dissolved in water, it's clear. Salt water, it's not going to separate out once you've dissolved it. The whole thing will pass through filter paper. Light can pass through it. And then an example, like we've been talking about, is a salt solution. So tonicity is also kind of like the concentration of the saltiness or the tonicity of a solution. So in this cartoon, it shows the different types of solutions. So if you have an isotonic solution, iso means the same, and they're saying tonicity means strength. So the strength of the solution. So in an isotonic solution, you have the same number of solute molecules as you do solvent molecules. So if there's 10 solute molecules in this cartoon, there's 10 solvent molecules. If you are a hypertonic solution, that means you have more solute particles than you do solvent particles. And if you are a hypotonic solution, that means you have less solute molecules than you do solvent molecules. So isotonic, hypertonic, and hypotonic are types of solutions. And then just to look at, this is a comparison of the properties of solution, colloid, and suspension. We've gone over this, but in terms of the test, you should know a suspension is a heterogeneous mixture. So hetero means different, so there's different things in it, and the example is blood. And in a heterogeneous mixture, for example, well, a heterogeneous mixture is an example of a suspension, and blood is an example of a heterogeneous mixture. But if you spin blood down in a centrifuge, large visible solutes will settle out to the bottom. So that's how you know that blood is a suspension. And this just shows mixtures versus compounds. If you are in a mixture, there's no chemical bonding between components. So you can see the red balls and these blue circles. They do not have any chemical bonds between them. It's a mixture. If you are a compound, you have a chemical bond between you. So remember, one of our favorite compounds is H2O. So there's polar covalent bonds in a water molecule, or water can be a compound or a molecule.